buongiorno. It's afternoon, actually.
It's afternoon. Yeah, I know. I know.
Okay, let me open it. PowerPoint. Here we go. And sorry again for the delay.
No,
Let's
we
change
are.
it.
I mean, we are okay. I mean,
We are in time. Yeah.
we are in time. We're in time. Okay, I will go first in Spanish and then in English. Yeah. First introducing the LATEPI. Buenos días a todos, a todas. Lo que estamos intentando organizar desde LatBrain, esta red LATEPI, la Red Latinoamericana de Epilepsia. Creemos con Luisa fuertemente que hay mucha capacidad en Latinoamérica que podemos unir y lograr que eso se transforme en un gran laboratorio internacional de epilepsia donde colaboremos y nuestros estudiantes se formen. Y de esa manera, esto es el puntapié inicial, pero bueno, siempre me gusta decir que la TEPI lo vamos a construir entre todos en la medida que podamos sumar esfuerzos, ¿no? Así que bueno, bienvenidas, bienvenidos. Esta es la primera charla disparadora de esta serie de encuentros. Y bueno, está abierto el email compartido para ustedes compartir anuncios, posibilidades de estadías y demás. Now I will go in English for Marco
I
and
think
for
I
any.
understood. I think I understood
Okay, okay.
95%. So
Oh, perfect.
I cannot speak
So,
in Spanish because it will be ridiculous, but I understand it.
okay, perfect. So the idea is that LATEPI, the Latin American Network of Epilepsy, will put together all the efforts that maybe we are doing in every country and put together technologies and students. We can move students not easily, but for sure it's cheaper than moving the student to Europe or to United States or Canada. And so the idea is that to put together all the efforts that we are doing in all the countries and try to make a big, international lab working in epilepsy in Latin America to put together our efforts. So that's basically the idea. The first talk is going to be, Marco de Curtis. Now Luisa is going to present you formally. And then we have another talk in the next month, and for sure we will have some other activities, easygoing activities like this one by Zoom to make all the people easy to come and to participate. So thank you very much, everybody, for coming, and you, Marco, for the talk.
Thank you so much. I'm very happy to present our first speaker, Dr. Marco de Curtis. Dr. Marco de Curtis was born in Milan, Italy. He is an MD, and thereafter he did a specialty in neurology. I'm very happy to tell you that Dr. Marco de Curtis has awarded with different grants and different awards. At present, he is author of 212 manuscripts published in Impact Factors in the Journal of Epilepsy. He is editorial board of different journals such as Epilepsia and Epilepsy Research. He has belonged to different scientific committees, especially from the International League of Epilepsy. I want to tell that he is co-founder and scientific committee member of the San Servolo Epilepsy course. That is a very important school for epilepsy that is carried out in Europe every year. He is director of the international course Bridging Clinical with Experimental Epileptology in San Servolo for different years, different editions. He was co-director of the international course on epilepsy in pathogenesis, founded in 2022. He is reviewer of different journals. He has been very active in the different commissions of the International League of Epilepsy. And also, Marco de Curtis has had a very important activity in the education through the world. Marco Thank you so much for to be our first speaker in this very important effort. Please go ahead.
Thank you very much. Thank you, Luisa. Thank you, Ramos. It is a real pleasure, and I'm flattered to be the first one to give this talk in this important new series of talks. And I like very much the idea that you're pushing through of having a virtual, let's say,
recording in progress.
Latin American laboratory where you can exchange people and experiences. Okay, so I will talk to you about focal seizures, focal epilepsies, and I will try to convince you that we do not know much about how focal seizures start, even though if you read the books, actually, there are several statements that are made which are based on all the work that has been done in the past. And the new knowledge that we have these days made things much more complex than the simplified version of epileptogenesis that we are used to trust, let's say, in a way. Okay, I'm a clinician and a neuroscientist, so I will start with some clinical issues, even if I understand that actually most of you are basic scientists. So we'll give some general concepts which are quite important, I think. One is this one of the new classification. Can you see it completely, or is this through the...
Yes, it is okay, Marco.
This is okay. Okay. Good. So this is the new classification of the International League Against Epilepsy. So if you work in epilepsy, even sorry. Okay, I have to close this actually. Do you see this red and green stuff? Okay, I move it down here. Okay. So I think it's very important to, even if you are a basic scientist, it's very important to know the basics of clinical epilepsy if you work in this field. And this is the new classification of the International League Against Epilepsy, which is very important. I think the most important part is this one on the right, because for the first time is clearly in the classification the evidence that there are many etiologies that are responsible for epilepsies. Epilepsy is not singular, it's epilepsies, plural, because there are many epilepsies due to many different diseases of the brain. So you can have a genetic problem, and this will generate a genetic disease with seizures, and this is what we call a genetic epilepsy. Or we can have an autoimmune response, for example, an autoimmune disease, and this will generate several problems throughout the body and also epilepsy. And this we call autoimmune epilepsies. But the message I wanted to give you, actually different causes generate different types of diseases which can or cannot be associated with epilepsy. When they are associated with seizures, when they are associated with seizure, we call them epilepsies. Okay, so there are two main types of epilepsies: the focal epilepsies and the generalized epilepsies. Most of the epilepsies indeed are of unknown etiology. So we see patients, we can identify a type of epilepsy, while we cannot say if it is focal or generalized. Focal epilepsies, they can be different. So we have some forms which are genetically determined, some other forms which are due to a specific known case, like malformation or trauma, tumor, stroke, and everything. So these are, again, different diseases that can generate focal seizures. And then there are focal epilepsies of unknown cause, which, again, represent probably 50%, even more, of the epilepsies. So I wanted to tell you this because to introduce the next step in my talk. So we know that we have some kind of epilepsies, like the malformation-related epilepsies, the epilepsies due to hippocampal sclerosis. And these epilepsies can be cured by they're pharmacoresistant, so they resist, the seizure resists to the pharmacological treatments that we have available, but they can be cured with surgery. Okay, this is an important thing, because if you go for surgery, then you can cure the patient, and you then have information about the tissue taken from the patient and from the recordings that are made during the tissue, during the preparation of the patient to the surgery. To go to epilepsy surgery, you have to rely on clinical data, so the story of the patient, the story of the seizures, on neuroimaging data, the MRI, the PET, and other things, and then also the video EEG data, which are very important because the EEG, as you know, is the main tool that we use to identify epilepsy seizures. So once you identify the site of origin of the seizures, which can be quite evident, like in this case, there is a lesion there, you can remove it. So this is the patient before surgery, after surgery. So a small hole is made to remove the epileptogenic areas. And the outcome usually of epilepsy surgery is excellent. Angle 1, there is a classification of outcome. Angle 1 is the best outcome. That means that after surgery, the patient is seizure-free for the entire life, or for a long time in their life. And as you can see, 70%, 71% of patients after surgery, they are seizure-free. Okay. For some specific cases of epilepsy, this 70% goes up to 85%. So it is very effective, epilepsy surgery. This is also a very important paper we contributed to. This is the list of the most common form of surgically remediable epilepsies in a series of almost 10,000 patients throughout Europe, in all the different surgery programs in Europe. And as you can see, hippocampal sclerosis is the most common form. These are a percentage, 36% of surgeries are done on hippocampal sclerosis. And then we have this ganglioma, or the DNET, so-called DNET, which also represent quite a high percentage, and then the focal cortical dysplasia. So this is a case of hippocampal sclerosis. This is a sclerosis. You can see it very well on the MRI. This is the sclerotic hippocampus. White means that it's sclerotic. This is reproduced in many of the animal models that you are using in your experimental work. This is an example of a DNET. So these are tumors which are congenital. So the patients are born with that, and usually they are not getting malignancy, but they are very epileptogenic. And then, unfortunately, it's covered. Sorry about that. This is a focal cortical dysplasia. So these are the three forms of most frequent tumor, epileptogenic lesions. And then there is also a very large amount of...
Achou que só porque você é bonito, você acha que você consegue tudo que você quer?
falou Marco?
Por Adriano?
Yeah, there is
Ele
an interference.
Ele não deixou o Adriano
Okay.
ingressar, não sei que droga que é. Não lembro
So,
agora, isso que eu tentar perguntar.
so somebody has
Fala
the
isso pra ele.
microphone on.
Mas é por isso que ele estava querendo ficar na casa.
Fez aqui.
Não tinha mais água, você já tinha tomado tudo.
I stopped myself because I have a background. I have a background noise. Somebody's talking. Okay.
Sabe que ele fez neurocirurgia, né? Ele é neurocirurgião hoje.
Quem, Marco?
O Adriano.
Yes.
Adriano?
Hello.
Please shut down your mic.
Ele se formou em medicina na França, lembra?
Adriano, o bonitão, não era medicina. Quando ele veio, quando ele estava com o ESP, ele não era medicina. Ele não era
Ele se
médico.
formou em medicina na França
Na
depois.
França. Ah, não, antes
Papa, you
contava
can do
com
it.
ela.
Foi lá
You
para
can do it
Marseille.
from
Não,
remote.
antes, o que ele era? Ele era... Isso que eu ia falar.
Então, aí ele chegou e entrou no terceiro ano.
Sim, também vinguei.
Fez neurocirurgia na Alemanha. Ele tentou vir aqui.
Sorry.
Okay, perfect. No problem. So actually, it took time to move this one so you can see it better. Okay, so these are the three forms, which most frequent form of focal epilepsies. But there is also a large number of cases where you do not see a lesion as clearly as in these cases. So you have a very focal epilepsy, but the MRI is normal. And this is also a very common case. So, okay, focal epilepsies. Now I wanted to show you what is the information that we have on the scalp EEG from these patients. Most of the time, you have a seizure. This is an EEG, a scalp EEG, so recording from the surface of the scalp, and this is a seizure. Okay, at the very beginning of the seizure, sometimes you don't see very strong, a big activity as it is here, but you can see some low voltage, fast activity. Okay, and this is seen even better when you record from inside the skull of the brain. And this is done, for example, in those cases where you have a very focal epilepsy, very localized, but without any structural alteration that you can see with the MRI. So in this case, you can make an hypothesis, yes, the lesion probably is, sorry, the epileptogenesis zone probably is in this area, and so you can implant a depth electrode like is shown here, several depth electrodes. This process is called stereo EEG recordings. And then you record for 1 week, 10 days from the patient. You try to capture seizures, and then you will see that actually individual spots here will localize, help you to localize where the seizures are generated. This is a very important procedure that actually is very useful to localize the epileptogenic zone. I'm showing here, for example, this is a case of a patient, and this is the onset of the seizure. And as you can see, it's very localized. This is a recording from different electrodes. And then the seizure spreads to different areas, and then it's ending, like in this case. So this is the type of recording that was made. So with several electrodes, each electrode, as I showed you before here, has different contacts. So you can record from up to 150 to 100 recording sites. So this is something beautiful because it gives you the chance to see the epileptiform activity from the real patients, so from patients, so beyond any animal model. And if you look at that, actually, again, also from inside the brain, so very close to the seizure onset zone, at the very beginning of the seizure, you have this kind of low voltage fast activity. So not big activity like here, but a very low voltage fast activity. This is quite common. So we look at all the cases that we recorded from in collaboration with the group of Niguarda Hospital in Milano, the group of Laura Tassi and Stefano Francione and the Munari group. And we analyzed the several seizures. These are the numbers. And in 85%, 90% of the patients, we always see this low voltage fast activity at the very beginning of a seizure. And if you look at the spectrogram, okay, I went home for a while without sound. I'm sorry. Can you hear me?
Yes, Marco.
Yes.
Okay. So actually, you did not hear me for a while, right?
No,
only the last slide, Marco. It was fine.
Okay.
Up to the last slide, it was fine.
Okay. So these are the recordings from the percentage of the number of recordings we did in collaboration with the group of Claudio Munari Center. And we have the most common pattern that we see is actually this low voltage fast activity. And if you look at the spectrogram, actually you have these are the frequencies and this is time. You can identify a very localized spot of activity around 125 Hertz in this case, 100 Hertz in this case. And we identified two patterns, one like this, shorter and with this very abrupt change in the jump into the voltage during the recording. Another one, which is we call L because it's longer and usually is in the limbic system. Okay, so pattern P plus L and L. This is pattern P. L. This is pattern P. Most of the patients at the very onset of the seizure, they have showed this pattern. Okay, so 92 patients out of the 100. Okay, so low voltage fast activity is the biomarker of the onset of a seizure. And this has been shown by others. This is a paper of about 10 years ago that all make an overview of the different papers they published in Time. And again, low voltage fast is the most frequent type of pattern that has been recognized. And also, this is another paper, review paper, the ictal onset is mainly. Do you see this in front of the slides?
Yes.
Okay, now it's gone. Okay, so the low voltage fast, which is here, is the most frequent pattern that you can identify in human recording of patients with focal epilepsy. Okay, so focal epilepsies are many. Those which are cured by surgery are a subgroup of these epilepsies. And when you record activity from these patients, from the brain of these patients, at the very onset of the seizure, there is a low voltage fast activity. So this is what we know from the human epilepsy. Now we can reproduce this pattern into animal models, and then we can analyze the activity of the animals, or we can take the brain of these animals into slices or do cultures and study the channels and the current and so on to verify what are the mechanisms that generate this human epilepsy pattern. So we can see it in this way. So this is the low voltage fast activity recorded in the patient. We can reproduce it in different animal models. For example, we worked a lot on this isolated guinea pig brain. So we can reproduce a similar pattern, low voltage fast, and then the rest. They're also in slices you can do the same. And then we use this model to analyze what are the determinants of these degenerative seizures, this pattern. Okay, that's what I will tell you about. What is in the book, though? What is written in the book? When you read a book on epilepsy, most of the time they say that you have a spike, the interictal activity. This spike is due to the depolarization of neurons, the PDS, which is generated by glutamatergic activity and sodium and calcium currents. And then at the end of the burst, you have this inhibitory mechanism that reduce the excitability. And then when this activity is increased and enhanced, then you have a transition into seizure. Okay, so this is what is written on the books. Even a book, this is a quite recent book, but was what has been probably most of you also see as the mechanism of generation of seizures. But this work is based on studies that have been done a long time ago. This is a Jasper's book on basic mechanisms of epilepsies. And this scheme derives from experiments that have been done in different models in the 80s, but before, let's say, the 1990s. But still, this was what is the main mechanism for most of the people working on epilepsy. But we try to analyze that with this different approach. So try to mimic activity generated in human epilepsies, and what we found is something quite different. These are a paper that we published, the work is done by Vadim Gnatkovsky. We use these isolated guinea pig brains, so the brain is perfused with the solution and is maintained alive for several hours. And then we induced the seizure activity, this low voltage fast seizure. This is an extracellular recording from an electrode position in the extracellular space. And this is a recording of a neuron, this principal cell, this is a pyramidal cell, during the seizure. And as you can see, at the very onset of the seizure, the firing of the cell is stopped. So the onset of the seizure is characterized by a reduction in the firing of principal excitatory cells. And this is highlighted here. So the very beginning here in A, activity is wiped out. And then you have your regular firing coming back when the neurons depolarize. And then at the end, you have this bursting activity. So at the very onset of a seizure, we do not have a lot of activity in principal cells. We did more work, and this was done in the entorhinal cortex of this guinea pig brain preparation. And we recorded from principal cells in superficial layers, in deep layers, and also recorded from interneurons. And what we found is that actually at the very beginning of a seizure, low voltage fast seizure, so similar to the one that is observed in humans, most of the principal cells from superficial and deep layers are quiet. Actually, they shut off their activity, while the local interneurons, the GABAergic cells, they increase their activity. So we made a lot of these recordings. He collaborated with Massimo Avoli, who was the first to actually make the statement that interneurons may be responsible for the generation of seizures. And then we ended up with this scheme of how low voltage fast activity seizure onset is generated. So here are each line here is the firing of a principal cell. This is of course a cartoon, so it's not real recordings. And each line here is the activity of a single interneuron, so a GABAergic cell. When you have interictal spikes, you have the firing, the PDS, let's say, associated firing, and then the post-PDS inhibition. generated by the activation of interneurons. But during low voltage fast activity, you can have these heading spikes, and these spikes are generated by a strong activation of interneurons, while the principal cell, the excitatory cell, they are quiet. These changes in interneuronal activity induce this huge increase in the extracellular potassium, and this is responsible for the depolarization of all neurons. And then they resume their firing and they entrain the continuation of the seizure. And at the end, the bursting of neurons is very strong in all type of cells, both principal cells and interneurons. So this is what we observed as a correlate of the low voltage fast activity. So a completely different story compared to what we are used to hear or reading in textbooks. Okay, interneurons then are in a way pro-epileptic. Okay, in normal brain, interneurons are very clever. They tune the excitability of different cells, and they play with the principal cells to generate spontaneous activity. And as you can see, the axon of one single interneuron, this is interneuron in the dentate gyrus, this is the axon, is contacting several 100,000 neurons. Okay, so if this cell is behaving in the wrong way, so is overexcited, then this cell can actually entrain in its excitation a lot of other cells. This has been shown in a very nice paper actually 30 years ago. This group of Somoji, they recorded from two principal cells, neurons like this, and then they stimulated the Schaffer collateral or the region where the interneurons are, and they could generate an inhibitory response in both cells. And as you can see, this inhibition is then facilitating a synchronization of cells. So the activation of inhibition can synchronize neurons. So this is something that can be amplified at the very onset of the seizure. It is also very well known that actually the normal EEG activity, like this gamma activity or other beta activities that form the normal EEG brain activity, they are supported by the activation of the the GABAergic cells. So the GABAergic cells, like the busted cell in this case, they're very much entrained into the generation of the synchronous population activity, which is part of the normal brain activity. So this actually, the story that actually the interneurons are leading seizures is not completely new, because if you look at some of the old papers, like this one by the group of Schiff, he also showed, but it did not highlight this observation, that if you record it from a slice of the hippocampus, of the entorhinal cortex, and you apply a specific form of stimulation to induce seizure activity, the interneurons, these cells, are active before the principal cells. So again, this has been observed before our work. And this has been shown also in models, chronic models of epilepsy, these like the pilocarpine rat model of epilepsy. Karen Moxon recorded several neurons, interneurons and principal cells. And what she noticed actually is that before the onset of a low voltage fast seizure, which is the extracellular recording here, the neurons, the interneurons are very active, more than the principal cells. So it's not only in slices, but it's also in vivo. Let's go back then to humans. So this is the first work that report this new piece of evidence. This is a recording from a patient. This is a dysplastic region, as you can see here. They recorded with macroelectrode like this one, again, to identify the epileptogenic zone, and also with this multi-electrode array, they recorded up to 140 single neurons. So each dot here is the firing of a single cell. And what they see is that when the seizure starts here at time 0, most of the neurons, they stop firing during the low voltage fast activity here. And some of the cells increase their firing. So this is the first evidence in humans that indeed the seizure starts with a reduction of most of the activity of most of principal cells. This has been also analyzed by the group of Los Angeles with this, again, electrode for stereo G recording. And at the end, at the tip of the electric field, there was this sprout of single rods from which they recorded units in the epileptogenic zone. They were able to identify inhibitory cells and excitatory cells. And also in this case, during the low voltage fast activity seizure, they saw an increase in the activity of interneurons, while the principal cells were quite quiet during the low voltage phase. Then they become active during the seizure. So to conclude this part, so low voltage fast activity, which is the most prevalent pattern that is seen in humans, can be reproduced in vitro. We use foraminopyridine to induce this kind of pattern. Can be seen in animal model of temporal lobe epilepsy, and is also observed in humans. The fact that interneurons are very active has been confirmed at all 3 levels, so in vitro, in vivo, in animals, and in humans. Okay, so interneurons, GABAergic interneurons, could be the promoters of seizure activity. And that's what we analyzed most recently with in my group, and the work was done mainly by Paolo Scarmani. We had slices of entorhinal cortex. We put it in a dish, and then we recorded with a patch clamp electrode here from a single cell, and also with the field director electrode, which is very important out here. So we give foraminopyridine, we perfuse foraminopyridine in the bath, and we have an increase of the background activity during foraminopyridine, and then at one point you have spikes. And one of these spikes then is leading to the seizure-like event. So we want to analyze which type of cells is responsible of this pattern, okay? Because we know that interneurons are very active. Which kind of interneuron? So we're recording from different type of interneurons. This is again a slice, the entorhinal cortex, the patch pipette, the field potential pipette. If we use animals, mice that have inside a GFP fluorescence located in the interneurons, we can identify interneurons, and then we can patch them. Then we can characterize them electrophysiologically. We identify 4 types of interneurons, those that contain GABA and parvalbumin, GABA and cholecystokinin, and 2 types of somatostatin-ergic cells. There is a lot of literature on that, so we can identify these 4 types. And also we recorded principal cells or pyramidal cells. And then we looked which one of these were active or were generating the seizure. What we saw is actually interneurons, when you perfuse the foraminopyridine, are very active even before the seizure starts. This is the seizure. And we see them very active also during the seizure and also at the end of the seizure. So this confirms that actually interneurons are very active throughout the generation of the focal seizure induced in the entorhinal cortex by foraminopyridine. Principal cells were also active, but not as much as the interneurons. And this is shown here. So before the seizure, a lot of activity in all subtype of interneurons, alpha-bumin, CCK, somatostatin, and not much activity in principal cells. Okay, but what happens when we have seizures? Time zero is the seizure, so is this point here or this point there. When you have a seizure where the arrow is, what we saw is that all neurons, all subtype of neurons, interneurons, GABAergic interneurons, we continue parvalbumin, CCK, and somatostatin, they were all active at the very onset of the seizure. And the same for principal cells. Sometimes the principal cells were following, sometimes were active at the same time. So what this shows is that indeed, if we induce a low voltage fast type of seizure, which is representative of the human seizures, all neurons are doing the same thing. They are all entrained at the same time, regardless of their physiological function in the normal brain. So principal cells as well as interneurons are all very active. Okay, so what generates seizures is interneurons, is principal cells. I'm starting now to think that actually is neither one of the two elements. But at one point in 2023, I think, there was a meeting in Switzerland, a very nice meeting. It tells, if you like, epileptogenesis, sorry, 2002. You have to go to this meeting where all the news about how seizure starts, about seizure prediction, seizure detection is shown. And there was a symposium on who starts seizure. Michael Wenzel was defending the hypothesis of pyramidal neurons, Gilles Uberfeld interneurons. And then we wrote a paper, a short paper, that is published on epilepsy. And what we see that actually in different models and in patients, sometimes in some papers it is shown that glutamatergic cells are coming first or GABAergic interneurons. If you look at the recording from single unit from humans, and also in models of animal models. If you look at the studies done in vitro, most of the in vitro study says that actually GABAergic cells are more prone to generate seizures, so the first to be activated. Okay, so let's go back to one. I mean, I open a parenthesis now. So this is what we analyzed in the entorhinal cortex of in different animal models to mimic something that happens in the temporal lobe of humans. But we have different etiologies for seizure and different location of seizures. So this is another case, another type of region. This is the piriform cortex. And we did the same kind of experiment. So we applied foraminopyridine. And in this case, we have a completely different pattern. And the mechanisms that underlie this pattern do not relate to the hyperactivation of interneurons, but probably they are generated by the presence of unmyelinated fibers that generate big changes in potassium. So this low voltage fast, which is shown here as well, in some structure could be generated by interneurons, but in other region can be generated by other mechanisms. So picture is very complex. We should not just rely on one type of experiment and generalize that to all the brain. So if we see in the entorhinal cortex the involvement of interneurons at the onset of the seizure, in other region the pattern could be completely different. So brain is complex, life is complex, epilepsy is complex. It's not as simple as a balance of excitation and inhibition. Okay, what is our problem, though, we neurophysiologists? That we look at the world through the glasses of electrophysiology. So we see things and we can interpret things that we see. But maybe the generators of this pattern are not necessarily the same that express this activity. So neurophysiological activity is due to the activation of neurons, but it's not consequent that the onset, the initiation of this hyperactivation is possibly only due to neurons. Neurons express seizures, but they could not be, maybe, are not the cause of seizure. And that's actually what we ended up to think about by looking at going on with this experiment. This is not published yet, is on bioarchives. We are actually having problems in publishing it because it's not an easy idea probably to digest. So think about the previous conclusion. So we have interneurons very active at the beginning of the seizure, all type of interneurons, pyramidal cells which are also active. So there must be something that pushes all neurons to generate epileptiform activity that not necessarily is a group of cells, a group of neurons. Okay, so we look at different things. So again, we recorded from the entorhinal cortex, we applied for aminopyridine, we record from interneurons on principal cells. We see that actually you have these spikes, and then the seizure. These huge spikes correlate in all neurons we recorded from. These are principal cells, and the red are interneurons. They always correlate with a huge chloride current. So chloride current, which is mediated by GABA. But it's not a simple GABAergic activity. So we compare then this single activity of GABAergic release, which is this triangle, and compared to that, actually, the size of this chloride current was completely different. It was much bigger, much larger, as is shown here, longer. And all these big current, they correlated with the spike. Okay, every time you have an extracellular spike, you have this huge chloride current in interneurons and principal cells. So where does this current come from? We did a lot of experiments, try to analyze pharmacologically to dissect out this current, and we ended up looking at this. The name is X66 because I didn't want to show it in another meeting, but actually I can show it to you. The name is bestrophin. We have these bestrophin channels which are present only on astrocytes. This is a GFAP staining of the entorhinal cortex slice. This is this bestrophin 1 channel staining on astrocytes, and this is the comparison. So this channel is not on neurons, as you can see here, but it's only on astrocytes. So there is a huge release of chloride from cells, which is likely due to this the release of GABA from astrocytes. If we apply the blocker of this BEST1 channel, which is up here, the name is NCBP, I don't remember exactly. You get rid of the background inhibition. Also, you get rid of these big spikes. So there are a few papers that actually show that indeed, if you stimulate an astrocytes mechanically, you can induce a release of GABA. a chloride current, which is in this opposite sign here, which is associated with the chloride, a GABAergic release. So we know that GABA release is can be generated by astrocytes. Very few papers published more than 10 years ago. So again, we use this blocker of the best one, astrocytic GABA release. And then if we apply foraminopyridine again, we do see some activity, but we do not see seizure activity. Okay, so if you block the release of GABA from astrocytes, epilepsy, so seizure are not generated. We did the same thing in the isolated guinea pig brain. We induce foraminopyridine seizure, and then we apply this blocker of best one from astrocytes, and then we get rid of the seizure activity. Sometimes you have spreading depression, and then at the end, when we wash out this drug, you can have some excitability that is coming back. Okay, so what is the picture that we see? So that we have astrocytes. We have presynaptic glutamatergic and GABAergic terminals. This is a complex picture, actually. So in normal condition, you have a release of glutamate, release of GABA during background activity. Okay. When you apply foraminopyridine, this release is increased. So you have more glutamate and more GABA released. The glutamate is transforming GABA and is loaded in the terminals, which are very close to the synapses. And then for reasons that we do not know, probably there are some stretch associated receptors which are activated. The best one is one of these channels, which is activated by stretch. There is a massive release of GABA, and this massive release of GABA induces this huge chloride current associated with spikes. How then these changes in chloride are generating seizures is something we do not know, possibly because chloride does not stay in the cells, but is extruded with potassium and degenerated the changes in potassium that I showed you before. So in conclusion, then, we think that actually the pattern of seizure onset, focal seizure onset is mainly low voltage fast. But this low voltage fast can be generated by different mechanisms, which can be location dependent. And then these diverse seizure onset are not only due to an accident excitation. There is an involvement of different mechanisms, which are not just a repetition of the spiking activity. I did not talk about these last parts, but the main message is that actually epileptogenesis is a complex phenomenon which has to be reviewed completely and re-understood from the basis. To say just increasing excitation and decreasing inhibition is generating seizure is too simple nowadays. Because again, things are complex, the brain is complex, epilepsy is complex, and life is complex. So this is my group. Also of them are clinicians, although they are basic scientists. And I would like to thank the sponsors of this work and the collaboration with the group of Massimo Avoli, with whom we developed this idea of GABAergic cells being pro-epileptic. And also the people at the Cloud-Luminary Surgical Center, where I learned actually that seizures are not just big spikes, but can be generated by a very low voltage fast activity. And this actually changed my perspective with epilepsy. And with this, I thank you for your attention. I'm open to any question.
Thank you so much, Marco. I really appreciate your presentation. For a while, we are going to wait for questions. I have one. If interneurons are inducing inhibition, but can activate the hypersynchronization. Is it possible to believe that anti-seizure medications facilitating the function of the interneurons could facilitate also seizure activity? What do you think?
Okay, I think there is a big difference in terms of okay, so if you think about it, you have very strong drugs that are enhancing GABAergic activity, like benzodiazepine or phenobarbital. If you use a lot of that, you completely block all the postsynaptic activity you reduce the postsynaptic activity of cells, of inhibitory cells. Sorry, you reduce the activity of excitatory cells by enforcing an increase in the inhibitory function of GABAergic cells. And that's one thing. On the other side, when the interneurons become wild, they do things which are out of control. Still, if you apply their benzodiazepine, you probably get rid of the epileptiform activity. But this is not the same, okay? So as using drugs given chronically. For example, if you know, there are several drugs that have been generated in the 80s and 90s that increases the function of the GABAergic function. And most of these drugs were not a success because they were not very useful in epilepsy, like the pregabalin and the other enhancer of GABA. They were used for pain, for example. They were not very useful for epilepsy. So if you modulate strongly, you increase strongly inhibition, you get rid of all GABAergic activity. But if you modulate with the drug like pregabalin and others, then you don't have an effect on seizures. But again, I think that it's difficult very important to differentiate between the normal activity of interneurons and the abnormal activity of interneurons. When you have an abnormal activity of interneurons, it's very difficult to stop it. If not with benzodiazepine, but that's not a drug which is available for epilepsy, for chronic epilepsies.
Thank you. Is there any question? ¿Hay preguntas?
Luisa.
Sí.
Te pediría que vos hagas la pregunta de mi parte, porque yo en inglés no puedo. Pero me gustaría saber si en algún momento ha usado solamente bloqueantes de calcio o CBD para impedir esas crisis y si los slides con los que trabajó han sido de cerebros naturales, normales, o si ha usado slides de epilepsias refractarias.
Okay. Marco,
I think
you understood?
I think I understood. Yes, if I block synaptic transmission with cadmium chloride or cobalt, then you get rid of this activity. Okay, so it is generated by, it's not really generated by, so you need the synaptic release to generate this, because to fill the astrocytes with GABA, you need the release of glutamate from the rest of the brain. Okay. So if you block with the cadmium or with the blocker of calcium channels, then you block all the activities, but also these currents. And regarding the type of slice, we are now trying to do the same experiment in a specimen coming from epilepsy surgery, temporal lobe epilepsy surgery. We do see the same big current. Will be impossible to redo all the experiments that we did with the pharmacology because it will take hundreds of patients and it's impossible to do it. But we can analyze whether these currents are present in the human brain and if they are also abolished by this blocker of the best one channel. We did not use chronic animals because, again, when you put the tissue of a chronic animal in vitro, it does not generate seizures. So you always have to push seizures with some kind of compound, for aminopyridine, low magnesium, and so on. So yeah, we do acute experiments. We try to dissect out what the mechanisms of generation of the pattern that we see are. And then we try to make some kind of bridge with the human tissue and with the real epilepsy, which is still something that has been fueled by several other experiments that we need to do. I hope I answered.
Thank you, Marco. Dr. Leonor Lopez Meraz includes one question. She said, "Thank you, Dr. De Curtis. My question is whether you have used any GABA transporter inhibitors and avoided these chloride currents. I find the comment that epilepsy and seizures are very complex, very valuable."
Okay. Yes, we blocked actually the GAT1, and when we block the transporter GAT1, what we see is actually an increase in this current. Let me see if I can show it to you. So one picture is better than 1,000 words. So let's see if I can, if I have it here. Okay. No, I'm sorry, I don't have it. But if you use GAT1, so if you block the transport of GABA, then you block the transport also of the GABA inside the astrocytic terminals. So actually you increase the amount of GABA, okay? And then when the GABA is released in the extracellular space, the current which is generated is much longer because the reuptake is impeded. It's bigger, much larger, and also the rhythmicity of the spikes is modified. So if you block the reuptake, you increase this phenomenon, okay. And the spikes are bigger. We do not know if you use this blocker of the transporters, we have seizures because we never did that experiment. But we just wanted to see whether blocking the transporter, you can modify the dimension of this chloride current.
Thank you. Questions? More questions? Javier?
I have one, yeah. Thank you very much, Marco. I love this idea of astrocytes being, at the very beginning of the seizure. And I want you, to maybe to elaborate a little bit more, because we thought that astrocytes were involved after, the initial damage produced by the seizures, and now you are putting them, very early in the situation. What about, traumatic brain injury plus epilepsy or infections plus, astrocyte not being so naive in this process and being targeted by another, bad thing that can happen to the brain?
Yeah. So the experiment that we performed are normal brains put in vitro, and then we look at how they can generate some activity. So, but these are normal astrocytes, normal neurons, and so on. No, so not epileptic. I don't know if, for example, this best one channel is represented or is increased into in the brains of animals which are epileptic. I don't know if when you have post-traumatic epilepsy, you have an increase in astrocytes, and this goes in parallel with an increase maybe in the expression of this channel. We do not know. and the same for temporal lobe epilepsy. So we never tested whether this kind of activity is also responsible for seizures in the chronic animal models. When you have a reactive astrocytosis, and that is somewhat involved. So I have no response for that. So
thank you very much.
There is one question from Dr. Jerónimo Ausmendi from Argentina. Thank you, Marco, for your presentation. Does the resting potential of interneurons and pyramidal cells remain stable with epileptic seizures, or do they undergo a depolarization that may justify the effect of GABA?
Yes, so when you apply foraminopyridine, you increase the background noise of the system, and all neurons, mainly interneurons, but also principal cells, they increase their spontaneous activity, synaptic activity. So you increase spontaneous GABAergic and glutamatergic activity. And then when seizure starts, you have a huge depolarization of all cells. And I think this is driven mainly by the changes in potassium. So if potassium is increased from 3 millimolar to 8, 10 millimolar, as it happens and has been measured in seizures, then all the equilibrium potentials of all different currents is completely modified. The neurons depolarize, all neurons depolarize, the principal cells and the interneurons. And this will generate a period where you have very little neuronal firing, okay, but for interneurons. So I don't know, probably you're asking about depolarizing GABA and this kind of phenomenon. I think that depolarizing GABA, we never see a depolarizing GABA in our experiments. We see the hyperactivation of GABAergic cells, but we never see actually the depolarizing GABA occurring. Probably because we are using tissue which is normal tissue and not pathological tissue. But I don't know. I think that the depolarizing GABA has not been really demonstrated, definitely demonstrated by several studies. So there is this idea that GABA to be bad for the brain, it has to behave like glutamate, so you have to depolarize and so on. Why? Because we think that only depolarization is excitation. No, I mean, you can have depolarization, a reduction in excitation, and you can have a lot of activity of interneurons and an increase in excitation. So I think we are still too much linked to the idea that we have only excitatory synapses and inhibitory synapses, and they have to balance. But if you put this, if you think about the complexity of our brain, we have changes in ions whenever you have activity, changes in potassium, changes in pH, changes in release of neurotransmitters we do not want to think about because that will make things too complex. Adenosine, acetylcholine, and so on. So the activity is very, very, very complex. And we have to start thinking that making things too simple does not bring us anywhere. So you have to try to integrate as much as possible complexities and analyze complexities, because otherwise there will be no future development. So we still think that it is glutamate excite and GABA inhibits, or glia is only there just for feeding the neurons and things like that. That is too simple and is not any more valid. Okay, so we have to think that we have to start again to learn, thinking that actually things are much, 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 much more complex than we think, and we have to deal with that. But this is nice because for young people, they will have centuries to to go on to study things and complex things.
Marco, thank you so much. And I want from your side a final comment, because you are an excellent scientist combining basic and clinical aspects. And this situation is very, very important in order to understand how basic scientists can do the research in order to understand first clinicians. What do you suggest for our students and community in general?
Yeah. Yes, I mean, this is really opening something which is very close to my heart. So the idea that actually basic scientists, they must have some knowledge of clinical science, because if they're working in epilepsy, they must know what is epilepsy, the background, the basics, but they must know it, because this is a very important also to develop new ideas, to develop things which are updated from the clinical side. So clinical science is very, very scientific, and this is not very much appreciated from basic scientists. So basic scientists work a lot on the details, but then they miss the big picture. And the big picture is in the hand of the clinicians. So I think the basic scientists should spend at least a couple of weeks in their lifetime, or per 5 years, every 5 years, in the clinic, and understand what is the logic in the clinic. Understand that temporal epilepsy is not Dravet syndrome, is not a post-traumatic epilepsy. So this is very, very important. And we did this, as Luisa knows very well. We did together actually a pilot project where we asked basic scientists to go into the clinic. That was done for 60 people in different countries. Latin America, that was very active in this. Europe, not much in North America, because they make things difficult up there. In Africa, some places in Africa, some places in Asia. And we did this. So we asked basic scientists, we asked the clinicians to tutor basic scientists for a couple of weeks. And all basic scientists were really very excited by the experience, because they realized, probably for the first time, what epilepsy is. And clinical epileptology, they were also very excited, because they had questions from basic scientists that probably the average neurology will never ask. So there was a very reciprocal excitement in this thing. And I think I will promote it for the future, if internationally will be interested in that, to distribute this kind of training activity, which is locally made. So it doesn't require any money or anything, just some organization. And this, I think, will increase a lot the clinical translational value of basic science. And this can be applied to epilepsy, can be applied to a pathology, can be applied to any medical science. So the gap between basic scientists and clinicians has to be filled in a way. So it's very simple. So ask basic scientists to go to the clinic for a while. The other way around is more difficult, because neuroscience is much more complex than clinical epileptology in a way. But in this direction, from basic science to the clinic, I think is easy to do. It is very interesting for both environment to do it.
Marco, thank you so much. And I hope in the future you can give us another conference. Thank you.
Thanks a lot. It's been a pleasure. And I know the Latin America community now, because I've been at LAS a few times, and it's an excellent group, an excellent and variegated, let's say, group of people. And I think that I will be happy to give another talk or whatever. I mean, thanks a lot.
Thank you.
Bye.
Thank you.
Thank
Thank you
you.
so much. Thank you. Bye bye.
Ciao, ciao.